So let us come to the fluid mosaic model of the lipid bilayer. What it states is we have the lipid molecules, the lipids, and we have some protein which are incorporated into the layer itself, which we called membrane proteins. However, it has been seen that some of these proteins can span the whole membrane from inside to the outside of the cell, while some proteins they just stay submerged inside the lipid bilayer as if a glacier or an iceberg in a sea. These proteins are called peripheral membrane proteins. And we also have cholesterol molecule. So what cholesterol do is it gives rigidity to the lipid bilayer, or it makes it less fluid, less flexible. Apart from that, we also have sometimes different polysaccharides or carbohydrate attached to these lipids, or also to proteins. They usually stay outside of the hydrophobic core, and now here we can see an interesting thing, which is if you will look closer, there is a gap is more between the lipids here as compared to the lipids here. Now there is reason to it. The reason is that when we have these cholesterol molecules, as I said, they creates a less flexible portion in the lipid layer. So consider this is the whole lipid layer and we have some cholesterol molecules here. So they will make this portion less flexible, and we call these portions, like the ones we have here, as lipid rafts, as if some rafts floating inside the sea of lipid. And it has been observed that these lipid rafts are the portion of the bilayer where most of the transmembrane protein or any membrane proteins are localized. And so at first we have something called flexing movement. Now, as the name suggests, this is depending on the flexibility of the lipid molecule. Like this is an unsaturated flexible fatty acid chain, or it can be saturated too. And the thing is, it can act like a spring and fold onto itself, which will give rise to something like this. And this can be helpful while accommodating a small protein inside, maybe like we have this here, we can accommodate a protein here. Anyway, then comes the flip-flop movement. Now this one is one of the most interesting part. Here what happens, suppose I have this lipid molecule along with other lipid molecules here. It has been shown that sometime we can end up in a configuration where the two lipid molecules will just swap their position. It's just these two lipid molecules can swap their position anytime. Then we have the lateral movement. and this is something which can contribute to the movement of our lipid rafts. Suppose we have a lipid molecule here and we have some other lipid molecules here. Then the lipid molecule can move to the right or left arbitrarily. Considering here it moved towards right, the structure will look like something like this. So now consider that there is a bunch of lipids like that moving laterally, so they can take the whole raft with them from one position to other. Apart from that, we also have rotational motion where lipid can rotate along itself. So this is all about the lipid bilayer for now, and I won't talk much detail about the lipid layer. Rather, I'll focus on what happens inside the cell, because that